चलिए तो हम आज का एक और नया टॉपिक वी विल स्टार्ट नाउ टुडे अनदर टॉपिक दैट इज इलेक्ट्रोपोरेशन और उसके साथ साथ हम पढ़ेंगे liposomal ट्रांसफर. So let's start, and this lecture is in English language because there are many people who understand the English language, and I want to make this as a pan world so that everyone will get benefit from my lectures that are very deep and very I will try to give the complete knowledge in these lectures. So, if you are joining my lecture, please subscribe the channel and like this, and share this with the other friends. And if you feel that there should be some improvement in it, or if you feel that some live session should happen with us, so that we will also join it. So for that, you can also comment. Okay, now good morning, everyone, and today we are going to discuss electroporation mediated transfer, gene transfer, and liposome mediated gene delivery, and two important in vitro and in vivo methods. So we are studying in vitro and in vivo methods. One is in vitro and another is in vivo method. So we are studying one in vitro method and in vivo method, and this method is for introducing foreign DNA into the living cell. So we want to introduce the foreign DNA into the living cell. For foreign DNA into living cell. So first of all, we will look at the basic principle, the step-by-step procedure, the advantage and the limitations, and finally how liposome can be combined with electroporation to improve the gene delivery. So these are the basic steps that we are going to study in this lecture. And now we will see what is the gene transfer. What is the gene transfer? Define me what is the gene transfer and what is the purpose of this gene transfer. Now, first of all, I would like to share this. I want you to share this video and share this channel so that it will reach to many people. So let us share it simultaneously. And during that time, I request you to uh, comment below so that people will start joining it. And now, just a minute, I will share it. First of all, sharing will make it easy to reach other people. Then we will go on with our lecture. Okay, let me share it. Now, so what is the gene transfer? First of all, we will try to know what is the gene transfer. Gene transfer. Gene transfer. So, the first topic we will study the gene transfer. So, gene transfer means delivering exogenous DNA or RNA into the target cell. So, we have exogenous. What is exogenous? That is outside the cell DNA into the target cell. This is the target cell. So the gene transfer is transferring the exogenous DNA into the target cell so that the cell can express the encoded gene product and such as a protein or therapeutic RNA. So cell can express, it can express encoded gene product. gene product. So this is important thing so that we can get the desired gene or a therapeutic protein or a therapeutic RNA. So there are two broad categories. There are two broad categories through which this happens, gene transfer happens. So we will study each of them. What are the two broad categories? One of the broad category is that is viral method. That is viral method. So we have two categories. One is viral method. One is viral method using engineered virus, using engineered virus, and another is non-viral method. Non-viral method. Another is non-viral method. So one is viral method and another is non-viral method. In non-viral method, we have electroporation, and in viral method we have lipos. In non-viral method, another is liposome mediated transfer, dated transfer, and gene gun, gene gun, and micro injection. So these are all the methods which are considered as the non-viral. These are the methods which are considered as the non-viral. Now, if I think there is some glitch in this sound, internet is okay. Okay, internet is okay. Now, so we have understood that there are viral methods and non-viral methods. One that is viral method is electroporation, liposome mediated method, and gene transfer method. And another is viral method in which viral engineered viruses are used. Engineered viral methods are used. Okay. Okay. Now, today we will focus on the non-viral physical methods. One is electroporation and liposome mediated method. So first of all, we will study what is electroporation, and we will study the basic principle of electroporation. So electroporation is based on the principle that high voltage electric pulse can induce cell membrane to fuse. That means there will be the high voltage electric pulse, and it is a physical method, and where a brief high voltage electric pulse is applied to cells suspended in the solution containing DNA. That means there is a solution, and this is the DNA solution, and this is a target tissue, and it is the cell is input in between the two electrodes, and high voltage electric pulse is given. Now, these pores allow DNA to and what happens, because under the electrical field, the phospholipid bilayer, what happens to the phospholipid bilayer of the plasma membrane becomes temporarily destabilized, forming transient aqueous pores. So this becomes destabilizer. That is depolarization. Depolarization happens, and this causes it distability. So distability happens, and this distability, distabilized membrane creates pores. So pore formation takes place. Pore formation. Now, this electric shock can induce a cellular uptake of exogenous DNA and believe it to be via pore from the electric pulse and from the suspended solution. And it is simple, rapid technique for introducing the gene into the cell of various organisms, microorganisms, plants, and animals. So this is a brief diagram which shows that before the pulse, the ions inside the cell are dispersed, and then the voltage is introduced and electric field induces a voltage across the membrane. That means the positive charge comes inside, negative charge comes outside, and this creates a pore formation, and through these pores the DNA gets entered into the cell, and this is the way that cell heals with the gene or drug inside and after the pulse. Now, this very important point that you need to understand further. The pores allow DNA to enter and the cell by diffusion and to the cell by diffusion and electrophoresis along the electrical field. After the pulse stops, the membrane reseals itself. That means the pores are formed. These pores, these pores are resealed. They are resealed after pulse is removed. Pulse is removed. So this is important thing. That means resealing takes place after the pulse is removed, and this cell is now with the having the DNA inside it. It's having the DNA inside it. Electroporation can increase gene delivery and expression by 100 to 1000 folds. This very important point, that is 100 to 1000 1000 folds. 100 to 1000 folds. That means 1000 folds. It can increase the gene delivery and expression. Gene delivery and expression. This is very important. No, okay. And compared to simple DNA addition, because more DNA molecules are taken in the cell and a large fraction of the cell becomes transfected. Now, what is the steps? What are the steps in electroporation mediated gene transfer? What are the basic steps? So, because we are reading all of these topics in a detail, and you will not find any such detailed video anywhere in the world that who is going to teach this online free videos by giving the detail. So we will go through the laboratory workflow. What is the, if you are working in the laboratory, and how people work there, and how the biotechnology process takes place in the laboratory. First, cells are harvested. In which phase cells are harvested? In the previous video I told you that cells are harvested in the log phase. Cells are harvested in the log phase. And what is OD at that time? It is 0.2 to 0.4. 0.4 should be the OD. You will understand that cells are in the log phase. So after the cells are harvested in the log phase, they are washed in low conductivity buffer. Second is washing in low conductivity buffer. The Witty buffer, they are washed in low conductivity buffer. For example, low salt or sucrose based buffer to reduce the joule heating and arcing. This is very important. Now, the third is mix with DNA. Mix with DNA. That is a known plasmid, a known amount of DNA plasmid. That is DNA, known amount of DNA plasmid is added to cell. It is added to cell and cell suspension in an electroporation cuvette. Electroporation cuvette. So there is the electroporation cuvette, on both sides of which there are electrodes, and this known amount of DNA is added. Applying the electric pulse, the cuvette is placed between the two electrodes. So this, like in the previous diagram, you will see that there is the this cell is in the cuvette, that is called the electroporation cuvette. Electroporation cuvette. Okay, so this is electroporation cuvette. Now they it is sided by the two electrodes. It is sided by the two electrodes and connected to the electroporator. It is connected to the electroporator, and you will see in the previous diagram, this is electroporator. Electroporator, this is electroporator. Now, and it's very easy how I'm explaining this, that we are having the cells from the log phase of the culture, and then we put these cells into the cuvette, and we add the DNA into the cuvette, and then we start to applying the voltage through the electroporator, and the cells are present inside the cuvette sided by the electrodes. Now a short pulse of high voltage, typically on the order of 100 to 1000 volts per centimeter, that is 100 to 1000 volts per centimeter is given, depending upon the cell types, for milliseconds, not the seconds, but for milliseconds. Okay, now the pore formation and DNA entry. During this process, there will be a pore formation, and there will be a DNA entry. This transient pore, that is for a small period of time, is formed in the membrane, and DNA migrates through them to the cytoplasm. And after the pulse in the warm culture, now how resealing takes place? When resealing takes place, for resealing, the cells are put cells are put in warm culture medium. So the cells are put into the warm culture medium and incubated to allow membrane resealing. That is incubated to allow membrane resealing. Membrane resealing. Now, after the pulse, the mixture is diluted in the warm culture, and we will get the cells which are having resealed. If the plasma contains a selectable marker such as antibiotic resistant cells, are plated on the selective medium to identify successfully. So marker is also important. So it is important to have the marker, and if the marker is present, so we can identify. We can um, identify. If marker is present, we can identify. It is important to understand marker is very important all these processes. Now we will erase this. Now what are the other steps that are key factors that will affect the electroporation? What are the key factors affecting the electroporation? The success of the electroporation depends on the several experimental parameters. What are the key factors? What are the key factors on which the electroporation efficiency is affected? So first is electric field strength. Electric field strength. First is electric field strength. So first is electric field strength. If the electric field is too low, pore do not form. Yani no pore formation if electric field is low, if electric field is low, an optimal electric field is chosen for the each cell type, like mammal, bacteria cell, yeast cell, plant cell, even tissues like skin all requires a different settings. So for a different settings we need a different optimal electric field, uh, voltage per centimeter. Pulse during the number of phases. First is electric field, second is pulse. How much pulse should be? Pulse duration should be pulse duration and number of phases, number of pulses. How many pulses are given? Longer or multiple pulses can increase DNA uptake. This is important. If we will give the longer or multiple pulses, DNA uptake will increase. DNA uptake will increase. Are you enjoying? Please comment if you are enjoying it. So you will not get such a detailed information in any of the video. So this is very important to you to say that yes, we are enjoying it, and it will encourage me to make more and more videos. Instead of, if you will share it and comment that I have shared it, then I will be thankful to you, and I will give your name in front of the audiences that this person has shared my video. Now DNA concentration is also, so first is pulse duration, second is first is electric field strength, second is pulse duration, third is DNA concentration. DNA concentration. DNA concentration and purity of DNA is also important. And purity of DNA is also important. Purity is also important. Now, purity is also important. Highly purified or supercoiled DNA gives better transfection than degraded or contaminated. So, highly purified, it should be DNA should be highly purified or and supercoiled than a degraded DNA. Now, cell density and growth phase is also important. So, cell density. It also affects if it is, and growth phase. In which growth phase you have took the cells? So mostly we take the log phase cells, usually efficiently transfected than stationary phase cells. So this need to be understood. Now, what are the advantages of this electroporation? What are the advantages of this electroporation? What are the advantages of electroporation? Now we will study advantages. Advantages. Few of the advantages are there, and don't no need to worry. So advantages are work for wide range of cells, like this works for wide range of cells. This works for wide range of cells, and like bacteria cell, like yeast, like plant protoplast, like mammalian cells, even tissues such as skin and muscles. Such a long variety of the cells it can be used. So this is the first, that is works wide range of the cells. This electroporation works over the wide range of the cells. And now, other advantage of this electroporation is high efficiency. That means this is highly efficient. This is highly efficient, high efficiency, high efficiency. This is highly efficient. So multiple plasmids can be delivered simultaneously and relative simple setup. It has a relatively relatively simple setup.